Our hypothesis for the standard photosynthesis test will be that the solution with dissolved bicarbonate in it, with dissolved sodium bicarbonate, will have a much higher rate of photosynthesis. In other words, the leaf chads will rise faster because there is a much larger source of carbon in the solution, as opposed to the just water plus soap, which will have to rely solely upon the carbon in the atmosphere. And we do have heat sinks on top of the beaker, which blocks a lot of other incoming gas with the water. So definitely the solution with sodium bicarbonate dissolved will have a higher rate of photosynthesis. I'm not faring very well, but I'll try to speak as best as I can. So our materials, we're going to need some baking soda, which is sodium bicarbonate, which is NaHCO3. We're going to need some liquid soap. Keep in mind that it will be diluted. 2 plastic syringes without the needle, 10 milliliters or larger. Some living leaves, in this case we're using spinach. A hole punch, a couple beakers, a timer, a light source, stirrers, weighing paper, a scale, all that jazz. After you have your sodium bicarbonate plus DH2O solution, you're going to add approximately one drop of dilute liquid soap to it. This will wet the leaf because the leaf has a hydrophobic surface, and so when you wet the leaf, the solution will be able to be absorbed by the leaf and it will sink. First thing what we're going to do is we're going to prepare our solutions, our bicarbonate solutions. So we're going to add 1 gram of baking soda per 100 milliliters of distilled water. Make sure you're using distilled water. If you're using tap water, just don't. And so when you have 200 milliliters of water, you're going to have to add 2 grams of baking soda. You cut your leaf chads. We use 10 for each trial. You are going to load them into the syringe, and you are going to draw some of your solution, and you're going to create a sort of vacuum. What you are going to do is you're going to push out in the air, and then you are going to block the top of the syringe, and you're going to pull it down to create a vacuum. And what this is going to do, it's going to push out any excess air and replace it with water. And once the leaf chads lose air, they will lose their buoyancy and they should sink. Now that you have your leaf chads, which are completely absent of air, you are going to put them in their respective beakers, and you're going to put a heat sink over them, and you're going to put a light source over them. The heat sink could be, you should fill them both with distilled water, just to be sure. And the purpose of the heat sink is just to make sure the water doesn't heat up during the experiment, because that is not a variable we are testing. So for our standard test, we test one of our beakers has a DH2O plus sodium bicarbonate plus soap, and the other one is just soap plus DH2O. These are our results for the standard test. So clearly something went wrong with our water plus soap formula. For some reason 1 chad did indeed go up, even though there was no source of carbon present. So I don't think the atmospheric carbon is enough to make that difference. So I guess there was contamination in our experiment. I'm sorry. My best guess for the contamination is that we only used 1 stir for both solutions, and so we contaminated the H2O plus soap solution with a little bit, but a significant amount of bicarbonate ion, which led to the 1 chad being able to rise.